important technology that was developed after the results of Human Genome Project was the DNA fingerprinting technology. DNA fingerprinting technology. This is a technology that helps us to compare the DNA of different persons. We must remember here that the DNA of no two persons will be identical. The DNA of no two persons will be identical, except in identical twins. Only in identical twins the DNA will be identical. Otherwise, comparing the DNA of any other two persons, you will find there is a difference. And these differences are found in the non-coding regions. Regions which do not function as genes. In the non-coding regions, that is in the 98% of the DNA, hardly about 2% functions as genes. About 98% of the DNA does not function as genes. So non-coding regions, this part of the DNA is commonly described as junk DNA. The DNA in the coding regions, the DNA in the so-called genes that have been identified, will be almost similar, 99.9% similar between any two individuals. If there are differences, the differences lie in the non-coding regions, in the junk DNA. And these differences are seen in specific regions of DNA called repetitive DNA. Specific regions called repetitive DNA. What is this repetitive DNA? These are regions where specific sequences of nitrogen bases repeat several times. several times. For example, there may be a sequence AATCC in one particular segment of the DNA. This sequence, adenine, thymine, cytosine, this sequence may repeat in a given segment, may repeat any number of times, 5 times, 15 times, 50 times, 100 times, 1000 times, and so on. May repeat any number of times. Therefore, such repeats are called variable number tandem repeats. Variable number tandem repeats, commonly abbreviated as VNTRs, variable number tandem repeats. For example, if we are comparing DNA of two different persons in a specific region, in the DNA of one person, this sequence AATCC may repeat, let us say, about 15 times, and in the same region of DNA in another person, this sequence AATCC may repeat 20 times, 30 times, and so on. Therefore, analysis of that particular region of the DNA of the two persons clearly differs from each other, so showing the difference between the persons. Therefore, in DNA fingerprinting technology, which was developed by an English biologist by name Alec Jeffries, in the year 1985. In DNA fingerprinting technology, it is these VNTRs that is taken for identifying the DNA, identifying the persons through the DNA. So, portions of DNA are isolated. They are subjected to a technique called gel electrophoresis. There are wells in the apparatus used for gel electrophoresis where the DNA sample can be put, when the apparatus is connected to electricity, the DNA fragments move along the gel. So depending upon the tandem repeats, where there are less tandem repeats, the distance travelled is less. Where there are more number of tandem repeats, the distance travelled is more, based on which the identity of DNA can be established. So the fragments which show movement in the electrophoresis are then transferred to a nylon membrane. This is called blotting. Then using radioactive probes, using radioactive pieces of DNA, the bands can be identified. The bands can be identified. This is, in brief, the technique used in DNA fingerprinting. We will see one PowerPoint presentation for more detailed description of the technique that is used in DNA fingerprinting.